All right, welcome back. So last time we got into the carbs and the lipids, and so today we're going to focus in on our proteins and nucleic acids. So if you're following along on our course objectives, we completed 3.3 and 3.4. So we're going to start with 3.5 today. Okay. All right, oops, wrong way. Okay, so let's get into proteins. Now, if you remember, proteins from that Amoeba Sisters video was the CHON, right? So CHON. So they all have a nitrogen in it, which the lipids and the carbs don't have. And proteins are very, very important in our bodies. They're basically like the building blocks of our bodies. And they actually are more than 50% of the dry mass of most of the cells are protein. So very important component of your body. And because they're so important, they actually have such a variety of different functions in your body. If you're thinking, if you ever get like a cold, they're part of your immune support. The storage, they help store different things in your body, transport things throughout your body, involved with cellular communication, movement, and structural support. So proteins are vital for our function. We are basically a lot of protein. That's basically what we are. So life would not be possible without a special type of protein called an enzyme. So most enzymes are protein-based. I shouldn't say all enzymes are protein-based, but most enzymes are protein-based. So enzymatic proteins, their job is to act as a catalyst to drive a chemical reaction. Okay, so remember in the last lecture, we talked about dehydration reactions and hydrolysis reactions. And remember how those reactants, they eventually will react together if you're leaving them there for long enough time, but they really need an enzyme to catalyze or speed up that reaction. So proteins, enzymatic proteins, are necessary to help speed up those chemical reactions that occur. All right. So we're going to watch a video of just some different proteins. So here's an example of a contractile protein.
Proteins, represented as purple blobs in this close-up of an animal cell, are the most complicated molecules known. A cell contains thousands of kinds of proteins, which carry out a variety of functions. In most cases, a protein's function depends on its complex three-dimensional structure. Let's see a demonstration of each of the protein molecules shown here and learn about its function. Observe how function depends on protein shape and changes in shape. Structural proteins have many functions. Like tent poles and ropes, they shape cells and anchor cell parts. They may serve as tracks along which cell parts can move. They bind cells together, making organized units such as muscles, ligaments, and the tendons that bind muscles to bones. The silk of spiders and the hair of mammals are also structural proteins. Signal proteins include hormonal proteins that help coordinate an organism's activities by acting as signals between cells. For example, insulin, a hormonal protein secreted by the pancreas, signals an animal's cells to take in sugar. The hormone receptor is also a protein. Signal molecules bind to receptor proteins that can then relay messages inside a cell. Receptors are thus important links in the system of communication among cells. Some signal molecules are also proteins. Transport proteins carry molecules from place to place. The example shown here allows certain solute molecules to enter the cell. Hemoglobin is the transport protein that carries oxygen in the blood. Sensory proteins detect environmental changes such as light and respond by emitting or producing signals that call for a response. An enzyme is a protein that changes the rate of a chemical reaction without itself being changed into a different molecule in the process. Enzymes promote and regulate virtually all chemical reactions in cells. Storage proteins stockpile building components that cells can use to make other proteins. Storage proteins in seeds provide raw materials used by the developing plant, unless an animal eats them first. Ovalbumin, the main substance in egg white, serves as a storage protein for developing chick embryos. Contractile proteins, like the motor protein shown here, can move parts of a cell. Other contractile proteins, working together in muscle cells, can move a whole animal. Gene regulatory proteins bind to DNA in particular locations and control whether or not certain genes will be read. This allows cells to become specialized for different functions and respond to changes in their surroundings. The immune system makes defensive proteins called antibodies that bind to invaders, such as the virus shown here, and mark the foreign objects for destruction.
All right, so we saw a number of different protein functions in this video, including the defensive proteins, the contractile proteins, the hormonal signal proteins, and so forth. So proteins have a lot of functions that are vital for your body to survive. So here's an overview of the different protein functions. And so we just broke it down into 8 categories. The first one was that enzymatic proteins. Remember, those are the catalysts that help speed up the reactions that occur in your body. So here's an example of a digestive enzyme that's catalyzing. Oh, look, it's breaking apart a disaccharide through hydrolysis, right? So this enzyme is breaking it down into the monosaccharides. And so that enzyme's speeding up that reaction. Storage proteins, so they can be used to store important amino acids because those are the building blocks of a protein. And that could be very helpful in, say, a developing embryo, like a chick in the egg here. So the ovalbumin is an important source of amino acids for that developing chick inside that egg. Hormonal proteins, so those are, hormones are chemical signals that get released from one part of your body and to convey some sort of chemical signal to another part. So they're part of our communication inside of our body, and they, not all hormones are protein-based. We talked about sex hormones are steroids, but a lot of our hormones like insulin, those are protein-based, so they can be used as chemical messengers. Contractile and motor proteins, so those are involved in movement. Your muscles are made of 2 important types called actin and myosin, so they work together in order for your muscles to move. Defensive proteins, so those are involved with our immune response against foreign invaders such as bacteria and viruses. So they send out these little antibodies made from protein to help destroy. Transport proteins, so they can help transport substances into a cell and out of the cell, but they also transport substances within a cell. So they can be used for both. Here's an example of a transport protein embedded in the plasma membrane, so it's allowing certain molecules to enter the cell. We also have transport proteins inside the cell that help transport things to where they need to go. Receptor proteins, so those are located on the surface of the cell membrane, and they're there to respond to some sort of chemical signal. And then structural proteins, they're involved with basically structural support. Think of your hair. Your hair is made up of keratin, which is a type of protein, and that is, and the nails on your fingertips, those are made of keratin, which again is a protective source and a structural support. So let's take a closer look at what a protein is made of. So the monomer of a protein is called an amino acid or a peptide. So you'll see that terms interchange throughout the term. So amino acids and peptides are the same thing. They're the monomer of the protein. So the protein would be an example of a polymer. So amino acids, remember that word amino acid, we talked about the functional groups that are involved. They have an amino group and a carboxyl group, in addition to something else. Okay, so here's just a basic structure of an amino acid. You can see the carbons. It's a chon, right? So there's the carbons, the hydrogens, the oxygens, and the nitrogens present in the general form. So our amino acid here has on one end a carboxyl group, and on the other end has an amino group. So all of our amino acids look like this for the most part. So it's kind of like this is the backbone of it. And then what differs between the amino acids from one another is this, what we call the R chain. So this R chain or side group is going to determine the function of that amino acid. So if we take a look at these two amino acids, leucine and serine, notice the backbone is the same. You got the carboxyl groups and you got the amino groups. And then the R group or side chains are different. So that's going to lead to their function. And in this case, leucine is an amino acid that's hydrophobic. So what did hydrophobic mean? Water fearing. And so in that case, the side group must have nonpolar covalent bonds, whereas serine is hydrophilic, okay? So it's water loving, so must have some polarity to it, all right? So here's just an overview of the 20 different amino acids of proteins. Do you need to memorize this? No, but you just realize that all of the backbones are the same, so you can see that in purple. So every single one of these is the same. Okay, we all have a carboxyl group and a amino group, and then in the ends. And then the only thing that's different is the side group or side chain or the R group. Okay, so you can see that some of them are going to be hydrophobic because of their nonpolar side chains. Some of them are hydrophilic because of their polar side chains. So some of them like water, some of them fear water. Okay, and then some can be electrically charged, and so they can act as acidic or basic in a solution. So these are both examples of hydrophilic if they're going to be dissolving in an aqueous solution. Okay. So let's see how a protein is made. So the first thing we need to do is we need to take the monomers and link them together to form a polymer. So our polymers are also known as polypeptides. So a polymer of amino acids is a polypeptide. And how is a polypeptide formed? Just like a carbohydrate, polysaccharide is formed through a dehydration synthesis reaction, where we have the OH group from the carboxyl end of one amino acid unites with the H from the amino group end of another amino acid, and they undergo dehydration. Okay, so that's where they're going to unite, and they're going to be covalently bonded together, just like with our carbohydrates and our lipids. They have a special name for their covalent bond. It's called a peptide bond. Okay, so they're forming these peptide bonds between the carboxyl group of one amino acid and the amino group of the other. So these polypeptide chains, they can range from a few to more than 1,000 monomers. Okay, so proteins can be really small or they can be really big. So each protein has a unique linear sequence of amino acids. Okay, and then each end of the amino acid, so if we have this big polypeptide chain, remember there's going to be one end of it that's going to end with the carboxyl group. Okay, so that's going to be called the C-terminus or carboxyl end. And then the other end will be the amino end or the N-terminus. So all of our polypeptide chains. One end will, eventually have the carboxyl group at the end, the C terminus. The other end will have the amino end, or the N terminus. Okay. So we have polypeptides. Polypeptides, you can also, sometimes you see the term polypeptide and protein interchanging. And it's, for the most part, you can just sit there and use both terms. It doesn't really mean anything that different. The only thing that it can be distinguished between the two terms, so they're both considered the polymers. Polypeptides, they are unbranched polymers, and the proteins are biologically functional molecules. So sometimes they're made of just one polypeptide, okay, or maybe they're made of multiple polypeptides. So you can think of a protein as, it could be just a polypeptide, okay, or it can be multiple polypeptides. So it's that old, keep bringing back the saying that all polypeptides are proteins, but not all proteins are just composed of one polypeptide. They could be multiple. So polypeptides, they are those peptide bonds, or the amino acid bonds or peptides that can be 100 to 1,000 amino acids or peptides long. And then again, remember how they're linked together through a peptide bond. Remember with the carbohydrates, you had the monosaccharides, two of them form a disaccharide. Well, when you have two amino acids or peptides, they can form a dipeptide. So that's an easy way to remember. So in what way is the production of a dipeptide similar to the production of a disaccharide? What type of reaction do they undergo to form a dipeptide? Same thing as the disaccharide. You take two monosaccharides, you undergo dehydration, and you get the disaccharide. Dipeptide, you take two peptides, bring them together, remove a water molecule through dehydration reaction to form a dipeptide. They both form a covalent bond, except in the disaccharide you have the glycosidic linkage. In the dipeptide you have a peptide bond. Okay, so they're both, the monomers are formed united together with dehydration reactions. Okay, so what's unique about a protein, though, is that the way it's folded will determine its function. So a functional protein consists of, it could just be a single polypeptide or more than one polypeptide. And the way that it's folded, it becomes this unique shape. So if there's any chance that the folding is incorrect, it's not going to function the way it's supposed to. So it has a very unique shape to it that's going to determine its function. So you can see, here's just some different ways to look at a protein. Here you basically you can't really see anything. It's just all the molecules. But here is that polypeptide chain, and you can see how the polypeptide chain looks, and then how it's folded will actually determine how it is used. In this case, this is an enzymatic protein, so it's going to be catalyzing reactions. So it needs to have a special area on it to allow for the reactants to bind to it. Okay, so it has a very unique shape. So the amino acid sequence is actually going to determine the shape. So each polypeptide leads to the protein's three-dimensional structure. And so if there is a change in that amino acid sequence, then you're going to change the entire structure of that protein. So that's why it's very important for us to understand a protein structure, because it's going to determine its function. So if there's anything that's changed in that structure, then it's going to cause the protein to not function correctly. So here's an example of an antibody protein, so part of your immune response. And we want that protein to be folded a certain way so that it gets a certain shape to basically bind to a flu virus. So if it has that certain shape, then that flu virus will bind to it, and then the antibody protein can destroy it. But if there's a slight change in that structure, then there's not going to be the binding that takes place, and so they won't interact, and then the flu will take over your body and make you sick. So it all comes down to the structure of the protein. So the structure determines the function. And proteins have a very, very complex structure, but it's a unique way that it's folded. So we're going to go through the different folding ways of the completed protein. And it is actually just basically 4 different structures. So not all proteins have all 4 of these structures. Some only have the A, B, and C. So the first part is the primary structure. So the primary structure is the most basic structure. It's just the amino acid sequence. You can see that we have the amino acids in a polypeptide chain. They're all held together by a peptide bond. So that's it, first structure, primary structure. But then those amino acids, they start to interact as the amino acid chain starts to fold. They interact with the hydrogen ends of the backbone and the carboxyl ends of the opposing backbone, and they create these little hydrogen bonds. So we'll talk about that because it creates a spiral shape in some of them called the alpha helix, or a kind of pleated shape, which we call the beta pleated sheet. So we'll talk about that. And then, so we've created this amino acid chain. It just looks like little beads on a chain. And then we folded it to form either an alpha helix or a beta pleated sheet. And then we're going to fold it even more when we combine those R chains. So that would be the tertiary structure. And so that's where it gets even more complicated because the arc chain is going to come in. Remember, some of those arc chains are hydrophilic and some of them are hydrophobic. So the hydrophobic ones are going to want to be kind of in the center, and the hydrophilic ones are going to be on the outside. So they arrange themselves. And then that's a single polypeptide unit. So proteins can be made of one polypeptide, so that could be a completed protein. That's functional. Or we might have multiple polypeptides, and they interact together to form a quaternary structure, which is the 4 polypeptide subunits or a complete protein. So we'll go through these in detail. We're going to start with the Amoeba Sisters video just to give us a good overview, and then we'll get into the details a little bit more.
We all have things that challenge us, and for me, it is folding. Sheets, towels, shirts. Let's just say I invest a lot in anti-wrinkle laundry spray. Amazing invention. My issue with folding extends to paper too. I know foldables in the classroom can be a powerful way to organize concepts, but for me, it was the actual folding part that I tended to get stuck on. You may think of folding as a convenience, of a way to take something and make it more organized or condensed so it doesn't have to take up so much space. This is true, but in biology, folding can actually have a lot to do with function. We've mentioned how amazing proteins are. They can play so many roles. They can make up channels, be part of a structure, serve as enzymes for important biological processes, be involved with protecting the body, just to name a few. We've also mentioned that you are making proteins all the time in a process known as protein synthesis. But the conclusion of producing a long chain of amino acids doesn't necessarily equal a functional protein. There are modifications to a protein that often need to happen in order for it to be functional. By modifications, we can mean many things. It might be adding certain chemical groups, such as phosphorylation, something to definitely explore. But another important event to make a functional protein is, believe it or not, folding. But before we get into protein folding, let's talk about shape and why shape is so important. Shape and function in biology frequently go hand in hand. In our cell signaling video, we mentioned how protein receptors and the signal molecules that bind them can fit together so perfectly to start some type of cellular response. Or in our enzyme video, we talk about how enzymes, which are frequently proteins, have a very specific shape for the substrates that they build up or break down. When we talk about the way proteins are folded, we need to understand the different levels of protein structure because there are different ways of folding that can happen in the different structural levels. The first level of protein structure is primary structure. This is the sequence of amino acids that make up a protein. Amino acids are the monomer, which means the building block, of a protein. They are held together by peptide bonds. In protein synthesis, amino acids are added to form a polypeptide chain, and proteins are made of one or more of these polypeptide chains. Genes, which are made of DNA, determine the order and number of these amino acids. That sequence is critical to the protein's structure and function. In our mutations video, we talk about how one amino acid can be changed in sickle cell disease. Even a single change of an amino acid has the potential to affect a protein's function. We do want to point out each amino acid has a carboxyl group, an amino group, and an R group. An R group is also called a side chain. So even though we have them drawn here like a chain of circles, realize that each of these circles we're drawing is an amino acid like this. Next, we move on to secondary structure. Folding is really going to start to happen. In secondary structure, the sequence of amino acids that we mentioned in primary structure can fold in different ways. The most common ways are the alpha helix and the beta pleated sheet. And which one of those foldings the protein does depends on the amino acid arrangement it has. Both of these shapes are due largely in part to hydrogen bonds. Those hydrogen bonds can occur at specific areas of the protein's amino acids. Specifically, these are hydrogen bonds involving the backbone of the amino acid structure. On to tertiary structure. This is looking at more folding that occurs in the 3D shape of a functional protein. And a lot of this is due to something we haven't mentioned much, the R groups, also called side chains. See, the amino group and the carboxyl group are generally standard parts of an amino acid, although the R group found in amino acids can vary among different amino acids. That means the R group can define the amino acid and can make amino acids behave a certain way. For example, some R groups are hydrophilic. They like water. Some R groups are hydrophobic. They don't. And remember that proteins contain many amino acids, which can contain different R groups, and so different areas of the protein can therefore be impacted based on those R groups. When protein folding is going on, amino acids with hydrophilic R groups may hang out on the outside, while hydrophobic R groups, where are they? They might hang out at the inside part of the protein. The 3D shape is due to other interactions besides hydrophobic interactions. Ionic bonds, van der Waals interactions, disulfide bonds, and hydrogen bonds, all involving the R groups, can influence the folding occurring in tertiary structure. Something to explore. Now, when we've been talking about a protein, we've been talking about a polypeptide chain that has been folded into a functional protein. But proteins can be made of one or more polypeptide chains. And in quaternary structure, you are looking at a protein consisting of more than one polypeptide chain. Each of these polypeptide chains can be a subunit, and interactions between them, such as hydrogen bonds or disulfide bonds, can keep them together. Going back to folding, I know what you might be thinking. Who is doing this folding anyway? Are the proteins just folding themselves? Well, the interactions mentioned, like hydrogen bonds and R group interactions, are occurring depending on the protein's own amino acids. One reason why amino acid sequences are very important for protein function. But folding is far more complex than that, and there can be intermediate steps involved when a protein is folding. In fact, there's a phrase you can search called the protein folding problem to learn more about the questions scientists continue to explore regarding protein folding. Research has shown that proteins often have help in the folding process. Chaperonins, for example, are proteins that can help with the folding process. They have almost a barrel shape. Proteins go into them, and the chaperonin tends to have an environment that is ideal for the protein's folding, and this can help the protein be folded correctly so it's functional. Just wish I had something like that for my towels. All of these interactions we mentioned in primary, secondary, tertiary, and quaternary structure are paramount for a mature protein to have its correct shape so it can carry out its function. And that's very relevant. There are many diseases that are related to protein misfoldings. Check out some of our further reading suggestions in the description about that. One last thing we haven't mentioned. Each protein has an ideal environment for functioning, which might include a certain temperature or pH range. If the protein is exposed to something outside of its ideal temperature or pH range, Exposed to high heat, for example, you can disrupt the interactions that we had talked about taking place at the different structural levels. This can denature the protein, which disrupts its shape. This prevents it from functioning correctly. And depending on what caused it to be denatured, sometimes you are interfering with many levels of protein structure. Sometimes it's just one or two levels. Sometimes denaturing a protein may be reversible, but in many other cases, it's not. The environment that a protein is in definitely matters for its functioning. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
Okay, so that was a great overview of protein folding. So let's go ahead and take a closer look at it. All right. There we go. Okay, so let's start with that primary structure. So we're going to go through all four structures and then just remind us how each folding occurs. So the first structure is just the primary structure, which is the polypeptide chain. So we see just the chain of amino acids all lined up, and remember they're held together by that peptide bond, which is a covalent bond. Okay. So that is just the first part of the protein structure, so the primary structure. All right. Next, we're going to start folding. Okay, so the second structure involves weak hydrogen bonds between the backbone of amino acids. Okay, so remember, here's the backbone, and then you have the R chains. Okay, so in the backbone, this hydrogen atom might start interacting with the nearby oxygen, or a different, probably a nearby oxygen from the carboxyl end of it, and form a hydrogen bond. And so the way that that happens, it might just be a simple folding, and then they have the interaction between them, so you get these beta pleated sheets, or it might be a twisting motion where you get this alpha helix. But basically it's the same thing, where you're taking the hydrogen and you're creating a hydrogen bond between an opposing oxygen group. So it can either form this helical shape or a beta pleated sheet shape, right? So hydrogen bonds are maintaining that secondary structure. Okay. In the tertiary structure, we involve the R group. So remember, the R group, or the side chains, they start to bond with nearby, structures as well. So the interactions between those R groups will start to change the shape even more. So instead of just having the helical shape or having the folded shape, they're going to start to build this tertiary structure. And that structure, a lot of it has to do with what the R chains are. So in the video, it talked about the hydrophilic and the hydrophobic R chains. So the side chains for the R groups. So if it's hydrophobic, it might want to be, inside the protein instead of hydrophilic on the outside. But that's not the only way that they can interact. So there's also ionic bonds that can occur, and the van der Waals interactions and so forth, and hydrogen bonds as well. But mainly we just think of it as these hydrophobic or hydrophilic. But it's starting to change the shape even more of that polypeptide chain. Okay. Now, this right here, this tertiary structure may become a protein, a fully functional protein. Or we might have multiple of these polypeptides that look like this coming together to form a quaternary structure. So a quaternary structure is when we have multiple polypeptides forming a functional protein. Okay. So here's an example of a quaternary structure of a protein called hemoglobin. So hemoglobin is a protein that's found in your red blood cells, and it has 4 subunits to it. So you can see 2 of the subunits are the same, and then you have the other ones that are different. And then this is basically the structure that's part of your red blood cell to help oxygen bind to it because it's an iron-rich structure. Okay. So let's take a look at the structure.
Most proteins are folded into a complex globular shape. Each protein molecule consists of one or more chains of amino acid monomers. The amino acids are linked by peptide bonds, so a protein polymer is often called a polypeptide. Because they are so complicated, proteins are usually described in terms of 4 levels of structure. Each protein has a unique primary structure, a particular number and sequence of amino acids making up the polypeptide chain. 20 different amino acids are used to build proteins. Theoretically, the various amino acids could be linked in almost any sequence, forming an almost infinite variety of different proteins. This illustration shows some of the amino acids making up the primary structure of a protein. The structure of a single generalized amino acid is shown below. The main backbone of every amino acid is the same. This is what forms the backbone of the polypeptide chain. It is the R group, which projects out from the backbone, that makes each of the 20 kinds of amino acids unique. Different amino acids have different properties that affect the folding of a protein. Thus, primary structure ultimately determines the shape of a protein, which determines its function. In most proteins, parts of the polypeptide chain are coiled or folded, forming twists and corrugations. This is secondary structure. The turns and folds of secondary structure contribute to the protein's overall shape. One kind of secondary structure is the alpha helix, where the chain twists. Another is the beta-pleated sheet, where the chain folds back on itself, or where 2 regions of the chain lie parallel to one another. Secondary structure results from hydrogen bonding between atoms along the polypeptide backbone. Oxygen and nitrogen atoms along the backbone are highly electronegative, and when bound to hydrogen atoms, O and N have partial negative charges, while the H atoms have partial positive charges. These negatively and positively charged atoms are attracted to one another at regular intervals along the chain, causing parts of the protein to twist or fold back upon itself. Superimposed on primary and secondary structure is tertiary structure, irregular loops and folds that give the protein its overall 3-dimensional shape. The irregular folding of the tertiary structure results from interactions among the R groups of amino acids. Acidic and basic R groups ionize, and these positively and negatively charged groups may form ionic bonds. Polar forces also contribute to tertiary structure. Hydrophilic or polar R groups may hydrogen bond with one another, or turn outward and hydrogen bond with the surrounding water. Hydrophobic nonpolar R groups cluster on the inside of the protein, away from water. Tertiary structure may be further stabilized by strong covalent bonds between sulfur atoms in certain R groups. Some proteins consist of 2 or more polypeptide chains. The 4th level of protein structure, quaternary structure, results from the combination of 2 or more polypeptide subunits. Quaternary structure is in turn stabilized by the same sorts of attractions that stabilize tertiary structure. Hemoglobin, the oxygen-carrying protein of blood, is an example of a protein with quaternary structure. It consists of 2 kinds of polypeptide chains. 2 of each, a total of 4 chains, make up each hemoglobin molecule.
All right, so we can see, the complexity of the protein with the primary, the secondary, the tertiary, and the quaternary structures. So this is a figure directly out of your textbook that might be something that you want to tag for, just further exploration because it, tells us exactly the difference between the primary, secondary, tertiary, and quaternary. So we have the primary structure being these linear amino acid chains. Secondary structure involves the hydrogen bonds. The tertiary structure involves the interaction of the R groups or the side chains, and then the quaternary structure involves the 2 or more of those polypeptides. Okay, so you can see the example of something like collagen or the hemoglobin molecule. So here's a question for you. If a genetic mutation changes the primary structure of the protein, how might this destroy the protein's function? So what is the primary structure? So remember, the primary structure is the amino acid sequence. And the amino acid sequence is going to determine the function of that protein, because as it's folding, okay, when you have the amino acid sequence for the primary structure, that will determine the folding for the secondary structure, which will determine the folding for the tertiary structure. So one tiny change in the amino acids can destroy the protein's function because it makes it not shaped correctly. Okay. So here's an example of that with hemoglobin. Hemoglobin, here is the normal primary structure for hemoglobin, and of course it forms a fully functional protein which makes our red blood cells look like a concave disc. But if you have a slight mutation in that primary structure, just one single mutation that instead of a amino acid called glutamate, you have valine in there, that is going to change how the entire protein is folded. So you can see how it's folded now. It looks different from the normal one. And this folding causes the red blood cell to be a sickle shape, leading to sickle cell disease. So one tiny change in that amino acid sequence causes a big change in the function of this protein. And now you end up with a red blood cell that can't do its job. Okay. So proteins, we know, are very important, and we know that their structure is held together by all sorts of different bonds. So we have the hydrogen bonds, we have you could have a disulfide bond, you can have the R groups joining together if they're hydrophilic or hydrophobic, or they can interact and ionize and stuff like that. So our proteins are very structured in a certain way that they're very specific, and they this specificity is going to allow only certain things to bind to them and them to bind to other molecules in order to function. So here's an example of a protein called lysozyme, which is a protein found in your tears, and you can see that the folding is exact in a way that there creates a little groove. And that's going to allow it to bind. It's a type of enzyme, so it's going to allow for certain things to bind to it. So that particular groove, if that groove is slightly altered in some way because the protein is misshapen, then it's simply not going to work. Okay, so proteins, they must recognize and bind to some other molecule to function. And so that's why they have to have this specific structure. And we also know that that specific structure being held together by all sorts of bonds, if we have a breakage in one of those bonds, then it can cause a protein or polypeptide chain to start unraveling. And that's called denaturing or denaturization. So when we have a denatured protein, it's been exposed to something that causes the breakage of those bonds holding the protein structure together. And things that can cause that breakage of the bonds could be changes in heat, like what we saw in that example. Salinity, so the amount of salt can change it. pH changes can also cause the bonds to change. So those are some examples. There's other things as well. Okay. So think about, what happens in your body when you have a fever. So why do you have a fever? Why would your body purposely increase its temperature when you're sick? Well, part of it is to basically kill off the bacteria, because the bacteria has proteins. It's got proteins inside of it. And those proteins will begin to denature at certain temperatures. So it's basically killing the bacteria by causing proteins to denature. Of course, if our temperature gets too high, then we end up with heat exhaustion or even heat stroke, and that can cause our own proteins to start to denature, which is could be life-threatening. So heat can have an issue in denaturing our proteins. Changes in pH can have an effect. So, you know, if we have a protein that's more used to an acidic environment, suddenly there's a bunch of hydroxyl ions introduced to make it more basic. The concentration shifts, and so you end up increasing the pH, and that can denature the environment. Okay, so that it denatures the protein. Changes in salinities as well, so it can mess with those ionic bonds. So a denatured protein typically is biologically inactive, so it can cause some adverse effects to the body. So why does a denatured protein no longer function normally? Well, it's not folded normally. And so if it's not folded normally, the shape is different. And if the shape is different, then it's not going to function the way it's supposed to. So the shape is determining its function. Okay. All right, so we did the carbs, we did the lipids, we did the proteins. We're on to the final one, the nucleic acids. And remember the nucleic acids, if we remember the thing from the Amoeba Sisters video, the CHO, CHO, CHON, and CHOMP. This is the CHOMP. So there's carbons, hydrogens, and oxygens, just like the other three. There's nitrogens, just like protein, but then we have the phosphate group or the phosphorus involved. Okay, so let's take a closer look at our nucleic acids. Now, whenever I think of nucleic acids, I automatically think of DNA. So DNA, that's what's DNA. It's our genetic information. So it's going to store, transmit, and help express hereditary information. So that's the function of our amino acids. So our genetic information is stored on our DNA. And why do we have our genetic information? Well, that's the instructions to make things in our body. So it's instructions to make proteins, for example. So the amino acid sequence that we just talked about with the proteins, that specific amino acid sequence is determined by your DNA, okay? So inside your DNA, you have little units called genes. And those genes code for a certain protein or an amino acid sequence. So that gene is going to specify what type of protein is made in that particular area of your body. And what are genes made of? Genes are made of DNA or nucleic acid. So nucleic acids are the polymer made up of the monomer nucleotides. So nucleotides are the monomers of our nucleic acids. Okay. So an example of a nucleic acid would be DNA. RNA is another example. Okay. So let's take a closer look at that nucleic acid. So we have two types of nucleic acids. We have deoxyribonucleic acid, DNA, and then we also have ribonucleic acid, or RNA. So if you take a closer look at that name, deoxyribo versus ribo, okay, and then it's just nucleic acid. Deoxyribose versus ribose, those are just sugars. So it's made of a special type of sugar. We have ribose sugar in RNA, deoxyribose sugar in DNA, okay? And as far as the function, the role of nucleic acids, well, DNA has our genetic information. So it's gonna, whenever your cell replicates, undergoes cell division, we need to basically replicate the instructions too. So we need to have each one of our cells in our body has a copy of our genetic code, our DNA. And so we need to replicate that DNA. So DNA provides directions for its own replication for cell division. It also directs the synthesis of RNA, a special type of RNA called mRNA. And that's going to take the message, the messenger RNA, to allow for proteins to be synthesized. Okay, so that particular segment of DNA will code for, will synthesize a unit of RNA called mRNA that's going to provide the directions to make a protein. Okay, so that's, since we're using a section of DNA and expressing it, that's called gene expression. All right, so let's take a closer look. Now, I think I showed you this slide in chapter 1 as well, but basically when we have DNA is always in the nucleus of the cell, okay, in a eukaryotic cell. So it's never going to be in the cytoplasm of the cell. So it's always housed in the nucleus. And DNA is long. It has 100 to 1,000 genes on it. And genes are just sections of DNA that code for a specific trait. So if we have, this huge long DNA, but we want to code for a specific protein, we only need just a little section of it to be expressed. So the DNA is going to basically direct the synthesis of a mRNA molecule that contains just that segment that codes for that specific protein. Okay, so the DNA and the RNA are going to be synthesized in the nucleus, and the RNA now has that code to make the protein. So it can squeeze out of the nucleus and attach to a protein maker called the ribosome. So the ribosome is going to read that code and put the sequence of amino acids in the correct order to make the correct protein, so the protein will fold correctly. So gene expression involves DNA to RNA to protein. This is also known as the central dogma, by the way. So let's take a closer look at what nucleic acids are made of. So I told you already that the monomer of a nucleic acid is a nucleotide. So that means that nucleic acids, they can also be known as polynucleotides, or many nucleotides together. So the monomer is the nucleotides. The polymer could be called a polynucleotide or nucleic acid, either way. And so here is an example of the monomer. And the monomer has a very specific setup, just like with the proteins and with the carbohydrates. So in the monomer, we have phosphate, sugar, and a nitrogenous base. So we always have this phosphate group, that's the CHOMP part of it, the P part of the CHOMP. We have the sugar group, and then we have this nitrogenous base. Now the phosphate group and the sugar group form the backbone to our DNA or RNA molecule. The nitrogenous base will vary, okay? So we always have this phosphate group and this sugar group, but the nitrogenous base will vary. So we actually have 4 different nitrogenous bases in DNA and RNA. So in DNA, that nitrogenous base could be an adenine, or it could be a guanine, or it could be a cytosine, or it could be a thymine. So that basically the nucleotide is determined by what nitrogenous base is there. So this is an example of a thymine nucleotide because it has thymine nitrogenous base attached. All right. So let's watch a video looking at the structure of both DNA and RNA.
DNA and RNA structure. DNA and RNA are nucleic acids, polymers made of subunits called nucleotides. This is a close-up of one DNA nucleotide. A DNA nucleotide is composed of a nitrogenous base, phosphate group, and a deoxyribose sugar. One difference between DNA and RNA is the type of sugar their nucleotides contain. DNA contains the sugar deoxyribose, while RNA contains the sugar ribose. Ribose has one more oxygen atom than deoxyribose. DNA and RNA are composed of 4 different nucleotides, which differ in their nitrogenous bases. 3 of the 4 bases are the same in DNA and RNA. adenine, guanine, and cytosine. The fourth base in DNA is thymine. In RNA, it is uracil. The nitrogenous bases guanine and adenine each have two linked rings of atoms. They are called purines. Cytosine, thymine, and uracil each have a single ring, and these three bases are called pyrimidines. For convenience, the carbon atoms in a nucleotide's sugars are numbered, beginning with the carbon atom bonded to the nitrogenous base, moving around the ring, and up to the carbon that is bonded to the phosphate group. The 1' carbon is bonded to the nitrogenous base, the 3' carbon to the next nucleotide, and the 5' carbon to the phosphate group. DNA and RNA are polynucleotides, long chains of nucleotides. Polynucleotides are always assembled in the 5' to 3' directions. A covalent bond forms between the carbon at the 3' position of a nucleotide and the phosphate group at the 5' position of the next nucleotide. RNA usually consists of a single polynucleotide chain. DNA, on the other hand, consists of two polynucleotide chains. The two DNA chains or strands are oriented in opposite directions and held together by hydrogen bonds between the nitrogenous bases on opposite strands. Because of their sizes, shapes, and arrangement of polar groups, the DNA bases form complementary pairs. Adenine pairs with thymine, and cytosine pairs with guanine. The two polynucleotides in DNA wind around each other to form the familiar double helix. The bases can be in any order like the letters of the alphabet. However, the base sequence is significant. Sequences of bases called genes encode the instructions for the structure and function of an organism.
All right, so I have two cats here that are kind of interacting, so if you hear some weird sounds in the background, that's what that is. So we have, basically that video went over the DNA and the RNA structure. So now you have a better idea. You know, we have the DNA, which is a double helix. It's two DNA strands held together by their nitrogenous bases. And then the RNA structure is basically the same. It still has that same sugar phosphate backbone, but it's in a single strand form. So let's take a look at what's really different about those nucleotides. So all nucleotides have the same sugar phosphate, just the sugar might be different. If it's DNA, it's deoxyribose. If it's RNA, it's ribose. So that's going to be the same. Those nitrogenous bases are what determine the type of a nucleotide though. And each nitrogenous base has one or two rings that include nitrogen atoms. So if it's a single ring, it's called a pyrimidine nitrogenous base. If it's a double ring structure, it's called a purine nitrogenous base. And there are, for DNA, there's two pyrimidines and two purines. So the pyrimidines would be cytosine and thymine. And the purines would be the adenine and the guanine. RNA is going to be the same thing, except instead of thymine as a pyrimidine, it has uracil, which is also a pyrimidine. So it basically replaces thymine there. Okay, so there is those different possible nitrogenous bases. And the sequence of those bases are going to determine the code, the information. So here we have our sugar phosphate backbone. And remember the sugar phosphate backbone, they talked about the 5' and the 3' end, which has to do with basically we just count the carbons on the sugar. So here is our sugar. So in this case, the sugar, basically the way we read it is this sugar, it's a 5 carbon sugar. And we start with where the oxygen is first, and then we go clockwise. Okay, so the first carbon in our ring here is called the 1' carbon, and then you have the 2', and then you have the 3', 4', and then the 5'. So when we have two nucleotides linked together, the 3' end of one nucleotide will attach to the phosphate group of another nucleotide. Okay, so here's a phosphate group of one nucleotide. Here is the 3' end of that other nucleotide. Okay, so they're held together between the phosphate group and the 3' end of the sugar. Okay. The sugar and the phosphate group of a single nucleotide are held together at the 5' end. Okay. So if we're thinking about it, we have a 5' end of our entire chain, and then we end up with this 3' end. So when we're building a nucleic acid, when we're adding more nucleotides, we always add it to the 3' end because the 5' end is already occupied, okay? It already is attached to the phosphate group. So we add something to the 3' end, which is where the OH is, and then that would undergo the dehydration reaction to form the linkage there, okay? And then this also just summarizes, again, the nitrogenous bases. So you can see the cytosine and thymine and the adenine and guanine in DNA. And then RNA would be cytosine, uracil, and adenine and guanine. Okay. And then the other difference between our DNA and the RNA is the type of sugar. So if you look at the sugars, they're both pentose sugars. They're both, 5 carbon sugars. The only difference, look at that, is this one's deoxyed. It has one less oxygen group, okay? So there's an OH group here for ribose, and this one's been deoxized. It's got one less oxygen, so it's just got a hydrogen there. That's the only difference. Okay. So when we have multiple nucleotides together, we call it a polynucleotide, which is also the polymer. Okay, and how they are joined together. Again, we talked about that sugar phosphate bone joining the nucleotides together. When we are talking about DNA, DNA is a double helix. And since DNA is a double helix, it has one side of nucleotides on one deoxyribonucleic acid, and then there's another deoxyribonucleic acid on the other side. And the two are held together by hydrogen bonds between the nitrogenous bases. So here is a cytosine nitrogenous base of one strand of DNA, and it's going to be bound to the guanine group of another nucleotide on an opposing strand of DNA. And it's going to be held together by a hydrogen bond, right? So that's why I keep referring to the sugar phosphate groups as the backbones, and then the nitrogenous bases are like the rungs of the ladder here. Okay, so let's watch a video on that.
DNA consists of 2 complementary strands of nucleotides twisted together to form a double helix. In this space-filling model, each atom is represented by a small sphere. The yellow and red phosphate groups make the 2 sugar phosphate backbones easy to spot. They're on the outside of the double helix. We will highlight the backbones in blue. Now let's look at the crossbars connecting the backbones. Each crossbar is a hydrogen-bonded pair of bases, one from each DNA strand. An adenine is always paired with a thymine and a guanine with a cytosine.
All right.
The double helix can also be represented by a ball and stick model. For simplicity, a ribbon model is often used, with ribbons for the sugar phosphate backbones.
Is it done? Okay, it looks like it's done. Okay, so we saw, you have basically it's 2 DNA strands held together by that hydrogen bond between the nitrogen base, or nitrogenous base. So RNA is a little different because it doesn't have this double helix. It's just one single strand. So that's one difference between the DNA and the RNA. But that RNA molecule can bind to a DNA strand and held together by a complementary pairing. So in DNA, A always binds to T. Let me write this down. Do I have it written down somewhere? I guess I don't. DNA, DNA, double helix, right? So we have our sugar phosphate backbones. That's just kind of like the sides of the ladder. And then the rungs of the ladder are the nitrogenous bases. So if I have an A, it's going to hydrogen bond with a thymine of the opposing strand. Okay, the cytosine will bind to a guanine of an opposing strand. Okay, so this is basically the C's always bind to the G's and the T's always bind to the A's. Okay. Now in RNA is single-stranded, right? You still have that sugar phosphate backbone, but you only have a single strand. So you have adenine, you have cytosine, you have guanine, and instead of thymine, you have uracil. But RNA is synthesized off of a DNA strand. So if I have this DNA strand here, let me just break it off. So here's the DNA, and it read adenine, cytosine, guanine, thymine. And let's say I'm using this DNA strand to synthesize an RNA molecule. So to make my RNA, I know that the adenine from the DNA is always going to bind with thymine on DNA, but it also binds to uracil on RNA. So uracil nucleotide will want to bind with that adenine. This cytosine will want to bind with a guanine. Okay, cytosine wants to bind with guanine, and then we have this thymine, so an adenine will want to bind to it. And so we build this RNA strand. So this is now RNA using this DNA template. Okay, so we have that complementary pairing that exists. So just remember, we always replace on RNA the T with a U. Okay, but otherwise it's going to be the same. The A binds to the T or the U, C binds to G. Okay? And then as far as the function of our nucleic acids are going to be involved with our genetic information. So remember our DNA, all of our genetic information is housed on DNA. RNA is going to be used along with DNA in the production of proteins, so it aids in protein synthesis because the RNA carries the code from the DNA, that the code being the instructions to build that polypeptide chain. And then the last thing that the nucleic acids can be involved in is cellular energy, because we have something called ATP, which is considered a nucleic acid. So ATP stands for adenosine triphosphate. So adenosine is very similar in structure to like adenine. And then we have these 3 phosphate groups, okay? And so adenosine triphosphate is going to be involved with a reaction that will release energy. And we'll get more into this in what was it, definitely in chapter 7. So that will probably be in like week 5 or 6. All right. So those are the functions. And then just some information about our nucleic acids. So our DNA is housed in every single one of our cells inside of our body. For the most part, not all cells have it, but a complete set of DNA is found, for example, in your skin. So I can take one single skin cell and have your blueprint to make another one of you. And so we've had this technology where we can take the information and figure out the genetic code to build a human. So that's called the Human Genome Project. So that was effectively completed in the early 2000s, where we were able to sequence the entire genetic code of a human. It took a long time to figure that out because there's like over 1 trillion of those those nucleotides. So the first human genome took over 100 years, or excuse me, 10 years. It took over a decade to complete just that sequence because there's over 1 trillion of those A's and C's and G's and T's, those nucleotides. So our genetic code is very complex and very long. Luckily, we can complete our human genome in just a few days because of new, faster technology, and it's actually less expensive. So some of you may have had your genome digitized if you ever did one of those, like the 23andMe DNA kits or something like that. So when you are submitting your DNA for one of those kits, they're basically sequencing your genome, and you can actually download your genome if you want. It is just a bunch of A's, T's, C's, and G's in a certain sequence. And so they can look at that and see, particular areas within your genome for certain genes, and that will determine, if there's a chance that, you might have a mutation that might increase your risk of developing breast cancer. That's the BRCA genes, or things like that. So that might give you a clue into some health conditions and ancestry and so forth. So a whole decade just to get one person's DNA, and now we can do it in a couple of years, or a couple of days, excuse me. So the number of genomes that have been fully sequenced has generated a lot of data. If you think about, how much data there is, so, we need to have certain software that can analyze that data. So that's involved in a particular type of computer science called bioinformatics. And so it's computer software that can analyze that special biological data. And this also plays into this idea of genomics, which is analyzing large sets of genes. And that way we can actually compare genes to other genomes and for ancestry purposes. And we can also use it to look at between different species to see how closely related species are to them. So genomics is, a very important field of study because it can not only help us figure out where we came from, so our ancestry, it can also give us key into evolution, how evolution happened. And it can also be used in our everyday medicine. So if we can tailor our medicine to our individual genome and how our body would react to it. There's also an area of study that looks instead of nucleic acids, it looks at proteins. So that's a new field of study that we're also looking at as well. Okay, so DNA and proteins, they can be used as tape measures of evolution. So remember, our DNA is just a sequence of nucleotides, a long sequence of nucleotides. And it houses basically all of our genetic information inside that DNA molecule. And we pass that molecule on to our kids. So we are, genetically linked to our parents, and our parents are genetically linked to their parents, and so forth. We inherit half of our DNA from our mom and half came from our dad. And since we have that ability to pass on our genes or our DNA to the next generation, we can see similarities between each other, okay? So for example, you and your siblings have very, very, very similar DNA, which makes sense because you have the same parents, okay? So there's greater similarity in the DNA between you and your siblings than you and me, for example, okay? Even though we're the same species, because you got your DNA from your parents and so did your siblings, okay? And the same can be applied between two species. So if two species are closely related, they're going to share more DNA in common with each other than they would with another species. Okay, so for example, our DNA is 99.6% the same as a bonobo, which is very similar to a chimpanzee. So only, a very small percentage of our DNA is different. Enough to make us not bonobos. We don't look like them, but we're very closely related to them. But we, as humans, between you and me, we share 99.9% of our DNA with each other because we're the same species. So you can see how the closer we are related, the more we have in common of our DNA. So molecular biology can be used to look at our evolutionary kinship with other species. All right. All right, so we are done with chapter 3. We've gone through all of the macromolecules. Make sure that you are understanding those functional groups because they help determine the function of the different macromolecules. All right, so we'll go ahead and stop here, and I'll see you. The next lecture will be on chapter 4, which will be in the next unit.